Hi, my name is Andrea Olsen. I'm in year 11, a student at Seven Oaks. And just before we begin this presentation, I want you to try and answer this question. What is memory? So most people have heard the term before, am I right? But it's pretty difficult to describe it literally. And my aim today is to share with you how I discovered a specific type of memory, fear-conditioned memory, through an internship I did this winter. So I came to Seven Oaks in year 9, and I started off fully engaged in lessons and personal development. But I realized that if I wanted to specialize in a specific field and start learning and doing something that I want to continue throughout my life, I would have to start sooner, and I would have to begin thinking about the jobs that I wanted to tune into. So I began researching different jobs, what career paths people can take, and thinking about what do I really want to do. And I stumbled upon neuroscience. This is the study of our nervous system and the brain. The brain is a fascinating organ. It controls our thoughts, our feelings, our senses, our sight, movement, and even sleep and circadian rhythms. And not a single invention has been close enough to replacing the human brain, no matter how people are scared of some artificial human being being created who can take over the world and evil robots and etc. But the more important thing is that there are so many unresearched things in neurobiology, like how our unconscious really works and what sleep is and what dreams are. That's still a mystery. So I set out on this journey to help discover new things in this field, and memory is just one out of thousands upon thousands of others that scientists are still researching. After this presentation, you will have learned about fear-conditioned memory, how it's applicable in life, how real scientific experiments and research is conducted, and how this knowledge can be used for future memory-connected inventions. For example, memory enhancers like superlearning, quick and efficient memorization, and memory erasers, for example, for post-traumatic stress disorder sufferers who have had traumatic experiences and want to get rid of their past memories. However, the main things I'd like you to take away are, firstly, the mechanism of researching the brain through experiments, but also just how much uncovered ground there is and how big of a future neurobiology has thanks to this. So in November, December last year, I visited a laboratory in Moscow called the Neurobiology Laboratory of Kurchatov Institute. This is led by a famous neurobiologist whose name is Konstantin Anokhin. This man is truly genius. So he's dedicated his life to researching memory pathways, and he's conducting fascinating studies, which I am sure will lead to an incredible future for neurobiology. So here I did experiments, and I got general experience on how to conduct different research studies in laboratories. During the first days, I got acquainted with the lab, the materials, met the people who were working there, the mice in the vivarium, and I did preparatory exercises for my experiment. These included immunohistological staining, and I will explain these in more detail very soon, brain cutting on a cryostat machine, fluorescent microscopy, and working with mice. This is immunohistological staining. So the way it works is that you mark specific proteins or you add specific antibodies, fluorescent antibodies that will mark the proteins in the brain slices that you're analyzing. So why is this important? Well, knowing if proteins were made in general means there's been gene expression happening, and that also suggests that the brain has been actively working, creating new neural connections and making memories. So the way we know if protein synthesis has happened is the computer that you use for analyzing these brain slices will interpret the fluorescent light given off by the antibody markers that you've put on the brain slices. Next preparatory exercise is brain cutting. So, here's fluorescent microscopy, first of all. So fluorescent microscopy is actually interpreting those antibody markers, which I just mentioned, and creating detailed photographs of brain slices for computational analysis later on, such as counting the number of neurons, etc. Here are a few photographs that I took, and this is me working in front of a computer, choosing the correct brain slices that I want to analyze. And the third preparatory exercise is cutting on the cryostat machine. So we can't actually analyze the full mouse brain, as you can see the photograph here, because that way you can only reach a few and a limited number of neurons, which you want to analyze. And if you cut the brain in thin slices like I'm doing in the video, then you can analyze specific neural connections and lone neurons in a much more precise and better way. And finally, how could I not mention working with mice? So in this preparatory exercise, I learned how to handle mice correctly, carry them around, safely transport them, and learn when mice are used in experiments and when not, because you can't use mice for all experiments, as first of all, they don't fit them, and second of all, we try not to use living beings for a lot of the experiments just because it's unethical, it's not allowed by the ethical committee of neurobiology, and generally, we try to conduct as many experiments as possible in vitro, which means without using any live organisms, but rather the cells in a cell culture. So, and unlike many people thinking that using mice in experiments is cruel, well, in fact, it's not, because mice live longer when they're experimental mice. They're well cared for. They get fed. They get water, and they're only used for the most important processes and experiments, which you cannot, unfortunately, conduct without living beings, as otherwise you wouldn't be able to progress and gain further knowledge. Now let's move on to the experiment itself. So the research question was, how does the pre-exposition to a location affect the freezing response, which is fear-conditioned response, of old and young mice as a result of fear-conditioned memory? Let me explain. First of all, you pre-expose mice to a specific location. This means letting them walk around, sniff for some time, and just get acquainted with the experimental sort of cage or box that they're in. At the next stage, you give them an electric shock in that cage. And at the last stage, you put them in the box for a third time in a row, and you check, how does the mouse respond to being placed in that location? Does it remember that it's been shocked there before, or does it not remember and not associate this location with a negative effect? So the independent variable in this study is how long a mouse spends in the cage during pre-exposition, and the dependent variable is the freezing time, and the freezing time is basically their response to being scared. So if a mouse freezes, we know that it's remembering something, it's associating this location with a previous electric shock that it's gotten. So the outline of the experiment is this. You firstly do the experiment with mice. So this consists of taking old mice, marking each mouse with the color to distinguish between them, because it's quite difficult to see the difference between genetically identical mice. Then you separate them into the groups that you want to experiment in, sort of in separate boxes. And then you pre-expose them on the first day of the experiment. And let me remind you, this consists of letting them walk around for like 1 or 5 minutes in the cage. Then you place them back into the home cage. Next day, you put the mice back into the experimental cage and give them an electric shock to their feet for 2 seconds. And they spend an overall 10 seconds in the cage. And for those who are interested, the electric shock is 0.01 amp. So it's not a painful shock, it's simply a stimulus that surprises these mice. Then you place the mice back into the home cage, test them or post-expose them in the electrical stimulus box. So you don't give them an electrical stimulus this third time, you just test whether they remember being shocked there by looking at their freezing response or not. And then you do the very same thing with young mice, because we're conducting the study on both young and old mice. The next stage is extracting the brains from the mice and storing them in the freezer for -80 degrees for a few days, because you need the slow temperature in order for the brains not to decompose and not to break down. And the next stage is cutting the brains on a cryostat machine. As I've said, you can't analyze the entire brain at the same time. You've got to cut them, and you got to cut it, and you got to look at the specific spots that interest you. Next is the immunohistological staining, which is the antibody marking. Fluorescent microscopy, interpreting these antibody markers to see the site of protein synthesis, basically where neural connections have been established. And finally, you analyze these results on a computer by counting the number of neurons, and you can make analysis and evaluation of that later on. So we had two groups in this experiment, old mice and young mice. Each of these groups had a control group of approximately 5 mice out of the total 30 per group, so 60 mice in total. And the control groups were not electrically shocked, and thus they would show minimal freezing time, according to our hypothesis, during the testing of how much they freeze at the end. Here is a video of me carrying out this experiment, or part of the experiment. You can see these are the experimental boxes where the mice are getting an electric shock. In the video on the right, you will see how we give them an electric shock. So you can see that they kind of freeze up for a second and look for 3, 2, 1. There we go. It freezes. We can see that it's felt the electric shock and then it suddenly is like, whoa, why did I just get an electric shock? And here is me just carrying out the experiment itself, recording some of the data on a computer, analyzing the movement of mice. Now, let's look at the results. Old mice have a lower freezing rate than young mice. This was one of our results. Why does this happen? Well, we believe this is due to the unflexibility of the neurons in an old mouse's brain. They cannot adapt as well to making new neural connections just because their neurons aren't that good at creating good connections. So this leads us to the conclusion that older mice remember less. The second conclusion we made was the longer the mice was pre-exposed to the cage, so the longer it got to roam around, sniff a little bit, get acquainted with the box before being electrically stimulated, the better it associated this box with the electric shock it got. So we saw this from our results because the mice that spent 1 minute rather than 5 being pre-exposed to this location had a lower freezing rate. And we know that freezing is the response to fear. So we deduce that the mice that are freezing more remember being shocked there and establish better memory. So you might be wondering, but what's the whole point of this experiment? Why do this at all? Well, this does open up new windows of opportunities for further research on memory. If scientists know how the time an organism spends somewhere prior to experiencing a fear conditioning stimulus, some traumatic experience like an electric shock or war, for example, we will know how this affects the memory of the organism. So we can deduce ways into lessening the negative impact on the organism. This is also quite important for people who have experienced shocking or negative experiences. For example, this research can be used for people who are suffering from post-traumatic stress disorder in, for example, wanting to erase their shocking memories. And although such research, like this experiment, seems insignificant at first, well, you're kind of just like giving electric shocks to mice, looking at the freezing rate, and so what next? Well, this can be the foundation of larger studies. It can be used as citation. It can be used as evidence of why something works later on, which is what leads to the most incredible discoveries. And the lab I work at right now, Kurchatov Institute, is looking at how memory can be erased after a shocking experience, and even how we can enhance learning and memory capabilities using different methods. So the research I conducted could be used for later human studies on how memory can be strengthened and lead to things like large capacity and fast learning. And this could help increase the quality of life. So I'm glad you made it through this introductory presentation on neurobiology and memory, and I hope I've shown you just how interesting this sphere of knowledge could be. If you've got any questions or queries, feel free to email me. But to end this presentation, I'd like to read for you a small quote from a famous American neurosurgeon, Wilder Penfield. He once said, "The brain is the organ of destiny. It holds within its humming mechanisms secrets that will determine the future of the human race." And I'd like for you to leave with a thought that neurobiology is an exciting and rapidly evolving study. There are things we know we don't know. There are things we know, but there are so many things that we don't know yet. So just like there was centuries of physical and medical discoveries, now is the era of neurobiology. Thank you.